Today, my guest is Antoine Adamantidis. He's in Bern, Switzerland right now, where he's living and working at the University Hospital there in the Department of Neurology, where he's associate professor. He's also the director of the Center for Experimental Neurology. He's an expert on sleep. And so today we're going to learn from him a lot about kind of the cutting edge research of what's known about what's going on in the brain during sleep and the importance of sleep in things like memory and even things like eating behaviors. So, Antonio, could you give our listeners and viewers background on you? Where did you grow up? How did you become interested in neuroscience and sleep in particular?
Yeah, sure. Thanks for having me. I was born in France and raised and educated in Belgium. So I did a university degree in biology and actually in plant biology, in cellular and molecular biology. So, but I did a PhD in molecular biology, but not on the plant side, but on the animal side. And the goal of my PhD was to generate a new knockout mice for a receptor that was just identified at that time. And then it turns out that that receptor is involved in sleep, and here it comes. I became involved in sleep research by studying the sleep phenotype of this new animal model.
That's interesting. It's how science often works. You start working on one thing and then something pops up and leads you in a direction you hadn't anticipated.
Yeah, exactly.
So you mentioned the plants, and one question about sleep is if an evolutionary perspective, it seems like all animals I know about anyways sleep. And do plants sleep? They do have a cycle. I mean, right at night, these, what are they, stomata, and the leaves open up and they kind of breathe or something.
Yeah, exactly. I think there is maybe not sleep as we define it in human or in mammalian system, but they do show plants and almost all organisms on Earth show a cycle of rest and activity. And this is basically induced by the fact that the sun comes and goes every day, every morning. And that creates some circadian rhythm that all organisms follow and adapt to evolution. So yes, they do have some decrease metabolism during when the light is not present, so at night, and then become more active biochemically speaking during the day when there is light.
Now, is our body, our metabolism of our whole body, muscles for sure, and probably other organs outside the brain goes down during sleep. Well, what about the brain cells? Are they actually reducing their activity, or
so some of, I mean, it was thought a long time ago that sleep was a passive phenomenon in the brain, and so that brain activity would decrease and then all of a sudden we would fall asleep. That's actually not the case. It's been shown that many cells are strongly active during sleep and basically generate sleep. So that being said, although there is a lot of activity in the brain during sleep, that activity might be different than during wakefulness. And in particular, those cells and a lot of the cells in the cortex, for instance, enter into this bimodal states where they're in up states, active, and then quiescent states, inactive. And then this succession of states occurs at roughly 1 second period, with a 1 second period. And so that basically creates oscillation that we can record with EEG. We probably will talk about that later. So there are some theories out there that suggest that this oscillation state with up and down is somewhat less energetically expensive as wakefulness, where activity is much more high and stochastic and yeah. So brain is not silent, but it is still in a mode of energy conservation. For the first part of the sleep, right, the light sleep, or what we call the non-REM, non-rapid eye movement sleep, for the deep, the REM sleep, or the paradoxical sleep, the dreaming states, there the energy is, the metabolism is much higher. It's completely different than the rest of the sleep. So during these dreaming states, there's a lot of neuronal activity. Also at the body level, the temperature is not regulated the same. So there's very huge difference between those two sleep states already.
And so you say that there's increased activity during REM sleep? And is that brain wide, or is it only in certain circuits in the brain?
It's in certain circuits, and mainly the limbic circuit, so the circuit that control memory, emotion, reaction, and stress, reward. Those are definitely much more active. Some other brain areas are decreased. The activity is decreased.
So those brain regions that are involved in emotion and so it's pretty important to remember things that have an emotional context, like a stressful event, or even, I guess, something potentially that's pleasurable. Is there evolutionary theory on why that might be, that those particular regions are?
Well, yeah, I think it's we could put that in a larger perspective where innate or adaptive behavior, learned behavior needs to be somewhat consolidated, optimized every day, depending on the day, depending on the season, if an animal is looking for food, or depending of time of the year, or depending also of the environment, the niche, how much predator there is around. And I think this behavior are so important in terms of survival of the individual, but also of the organism, that they are probably extremely refined by the brain. So that means that they are during wakefulness, they are definitely strongly supported by complex neural mechanism, and during sleep there are now a large body of experimental evidence that they are also remanipulated, and we thought it was only consolidated during sleep, but some are also weakened, so decreased during sleep. And
yeah, so that kind of makes sense. As we go through our day, we're constantly receiving a lot of inputs. Right now you and I are talking, later on, whatever, we'll be eating, talking with other people, moving around. And, over days anyway, we won't be able to remember a lot of the specific details of, our visual or auditory inputs, but things that have an emotional context we'll remember. So, there's this idea that, and actually evidence, which I think you can talk about, that there's this consolidation of memories, like a replay, in some way, a selective replay of events that are important for our survival and future functioning, I guess.
Yeah.
Do you want to talk a little bit about how that might occur? Is it thought it's just simply kind of the neurons that were active when we were actually experiencing that event are again active and kind of strengthening the synaptic connections during sleep?
Yeah, so this has been described for several systems in the brain, and maybe one of the most famous one is the place cell in the hippocampus. So basically, the place cell in the hippocampus are cells that are strongly activated when an organism, an animal in the lab or a human is walking in the street. You have to think about a 2 or 3D environment, and there are cells that fire at very specific place when the animal walk down a labyrinth, or if we are walking in specific streets. And so every place is encoded by different assemblies of cells. That means that basically there is a mapping inside the hippocampus of the environment that we are navigating through, right? And this is how the brain oriented and orienting is orienting ourselves or us in this environment. And interestingly, what has been found is that this sequence of cells that really discharge in a very specific order when the animal or human go into a specific path is reactivated during sleep. And so this has been found in many different labs, experiments, species, and it's been proposed that that replay during sleep actually consolidates our representation of the space, of the path we were walking through. So this idea of reactivation of previous experience during sleep has been extended to different brain circuits. So it is true for the hippocampus cell that we just talked about, but this has also been shown for some cells involved in decision-making, the prefrontal cortex, in cells controlling emotion in amygdala or reward pathway in the midbrain. So scientists have been describing this reactivation more and more in many different models, and there's really a consensus that this reactivation during very short period of time during sleep is important for consolidating synaptic connection between cells that support this information encoding, and therefore leave a trace, a persisting trace in the brain.
Yeah, so in the animals, you can actually put electrodes in their brain, and at least, you know, and that's the kind of studies I think you're talking about, where they're recording with electrodes the activity from even individual neurons in the hippocampus. And then, so that's really interesting that they replay. Essentially, you have a map in the brain of your environment. And it makes, again, from an evolutionary perspective, it makes a lot of sense, because in general, animals they move their environment, they encounter certain food sources in one place, they may encounter a predator in another place. And those are, you know, food and avoiding hazards is a big thing for survival. Or they, you know, if it's a male, they may encounter some females over here or something.
Yeah.
Have to remember that. So
yeah,
that's really fascinating. Okay, so can you go into a little more detail on the slow wave sleep, the REM sleep, and because these are defined by recording with EEG and different frequencies. Can you kind of briefly summarize?
Yeah, so as you said, we can record sleep with electrodes, right? And that's been a gold standard method to characterize sleep in the clinic or in the labs. So we have those tiny electrodes that we can put either on the scalp in human or on animals, and then record the brain activity, so the activity of a large amount of cells that is in the vicinity of those electrodes across wakefulness and sleep. And then based on and what we see when we record those electrodes is basically oscillation. So we see those recurrent oscillation, and they come from the synchronous activity of a group of cells, again, near the electrodes. And interestingly, based on those oscillations on the EEG, we can define either, as you said, slow wave sleep or non-rapid eye movement sleep, non-REM sleep, and REM sleep or paradoxical sleep, which is the dreaming states. And basically, in the first one, in slow wave sleep, the activity is rather slow, 0 to 5 Hertz, for instance. There are some spindle in 10, 15 Hz, but that's pretty much most of the activity you will record from a scalp electrode. From the hippocampus, if you put an electrode inside the hippocampus, one can record sharp wave ripples. Those are very fast oscillation, but that occurs only during 1 or 2 seconds. And then they disappear. They just come and go. And those are the oscillations during which the place cells are reactivated. And so this is for the first sleep stage. The second sleep stage is REM sleep, or that is very characterized by what people used to call a reactivation of the brain, because the EEG looks very similar to brain activity during wakefulness. There's a lot of high frequency. It's not so much of those low frequency oscillation. And there's a typical theta rhythm, for instance, that we see. So this is 6 to 9 Hz. So really, with those electrodes, one can really quantify or characterize sleep in terms of quantity, how much of the non-REM sleep one gets, or deep REM sleep, or in the quality. How is the brain activity? How are those waves, the amplitude, the frequency during sleeps, and what is the quality of the sleep of one patient, for instance. So one thing to note is that we make this distinction between those two sleep states, non-REM and REM, or slow wave and REM. In human, there are actually 3 subdivisions of the non-REM sleep, 1, 2, 3, based on whether it's light or deep sleep, right? And it's called slow wave sleep because the state 3 is the state during which there is a lot of slow wave, those 1 Hz oscillation that we were talking about earlier, and that are predominant during that state. And then during the whole night, human have a sleep cycle of about 90 minutes. That means that the brain slowly goes from wakefulness to non-REM 1, and then 2, and then 3, and then finally goes to REM sleep. And the whole cycle from wake, non-REM 1, 2, 3 to REM takes 90 minutes. Where it's quite different in animals. In mice, for instance, a cycle is around 5, 6, 7 minutes. In rats, a little short, a little longer. And that's basically well described in the literature, that the length of the sleep cycle is directly correlates with the metabolic rate. So the higher the metabolic rate, the shorter the sleep cycle.
Now, during one phase of sleep, your muscles become almost paralyzed, right? Which phase is that?
So yes, this is something I didn't mention. I was getting there. This is during REM sleep. There is a muscle atonia. And this is an active process. So the brain is actively inhibiting the motor output. And this is the mechanism has been quite well described now in animals. There are several models of this disease. Not the disease, but the mechanism. And this is why that state has been called paradoxical sleep, is because the brain activity is very similar, although it's not the same, but it's very similar to the brain activity during wakefulness. It's highly active, high frequency, lots of neuronal firing, but the body is completely asleep, and that's why it was called paradoxical sleep. Brain wide awake, body wide asleep. And so,
I used to train and race what we call harness horses, standardbred. And horses, a lot of horses will sleep standing up.
Yeah.
Has anyone looked at, what?
So I think, yeah. They actually have a mechanism, and I think it's morphological in their articulation, where they can block those articulations to sleep while they are standing up. Yeah. And I think that also happens in some birds.
And sleepwalking in humans, dude.
So this is a parasomnia. This is we don't know exactly the mechanism, but there are some studies that suggest that there is a disconnection between the frontal and the parietal cortex, and this happens during non-REM sleep, slow wave sleep, not during REM sleep. Yet there is one disorder of REM that is called RBD, REM sleep behavior disorder, where human patients enact their dream. And so that's very often people who are they're completely asleep, but they can manifest very elaborate behavior, stand up, grab a bed table, a chair, books. They can be also very emotional, and it could be aggressive sometimes. And this is due to, we believe, in a lack of inhibition of this motor output during REM sleep. Interestingly, this is also an early, maybe not an early sign, but it is strongly associated with development of Parkinson's disease later on.
Is there any genetic and is there any genetic work on that to see if it runs in families?
Yeah, there are some genetic component, although not only, but certainly for many of the sleep disorder, there have been some genome loci that have been associated with those sleep disorder.
I guess as long as we're on sleep disorders, the one maybe some of our listeners and viewers have heard about is narcolepsy. You want to talk about that, and then I guess we can get into talking about specific brain regions and maybe focus a little bit on the hypothalamus, which is really important. So what's narcolepsy?
So it's a good transition, because narcolepsy is a sleep disorder in which patients have a perturbation of their intrusion of REM sleep during wakefulness, and sleep fragmentation together with hypnagogic hallucination when they go to sleep or wake up. But they also have, and that's the most predominant symptom, they have cataplexy. So when they are awake and when they are usually engaged into social context, if you tell them joke, for instance, they have this progressive lack of muscle tone, and they really fall on the ground or from their chair. So it can be very dangerous in kids. They can be very dramatic and for the patient, but also for the caretakers. And this disorder is as a genetic cause. Some cases are purely genetic, and there might be also autoimmune mechanism. So, back to what we just talked about, not all the sleep disorders are genetics. There are some autoimmune mechanisms, as this has been recently shown. But one of the important finding about narcolepsy is that patients with narcolepsy lack specific gene transcript in the hypothalamus. So the hypothalamus is at the bottom of the brain, and this is a brain area that controls, basically, we're talking about innate behavior. It controls most of the innate behavior, but also homeostasis. So that includes food intake, metabolism, stress reaction, fight and flight response in animal, reproduction, sleep, and wakefulness, and so forth. So it's been found that the narcoleptic patient don't have the gene transcript for a gene that is called orexin or hypocretin, because it's been discovered by two different groups. And this gene is expressed only in cells that are located in the lateral hypothalamus. That's the only place in the brain where you can find those cells. And patients with narcolepsy lack this gene transcript. We don't know if the cells are missing. There might be indication that the cells are still there, but this is still an open question. But basically, that's has been a huge discovery in the field of both in sleep medicine and experimental sleep research. And I think the breakthrough prize this year was given to Professor Emmanuel Mignot at Stanford and Professor Masashi Yanagisawa and Tsukuba Japan, because they both discovered this link between hypocretin and orexin gene and narcolepsy. And
so this is interesting. So these these neurons in the hypothalamus that are producing this orexin hypocretin, they must be communicating with a lot of different brain regions if, you know, they're having this dramatic influence on sleep stage.
Yeah, exactly. So I've been involved when I was at Stanford as a postdoc with my supervisor, Professor Luis de Lecea. We were studying the role of those cells, those orexin hypocretin neurons, on arousal and wakefulness. And those neurons, although they're located only in the hypothalamus, they send projection almost in every part of the brain. And they're not the only one. There are a lot of cells in the brain that are that have this diffuse projection pattern, and they are usually identified as a neuromodulatory system of the brain because they modulate many different functions, including sleep and wakefulness. And hypocretin orexin cells in this case are known to modulate arousal and wakefulness. And the experiment that we did at that time on this system was that we selectively activate those cells with a technique that is called optogenetics. We can talk about it later. But and then when we activate those cells during sleep, that increased the probability of wakefulness. So that was the first study to establish a causal link between activity of those cells and transition to wakefulness.
Now Antoine, so you were at Stanford for a while, right? And did you work with Carl Dieseroth?
Yeah, at that time we developed that technology that is called optogenetics together with him, and to really study those wake arousal neurons in the brain.
And I see he recently got the Lasker Award, which which is historically, not always, but in many cases, someone who gets the Lasker Award will go on later to get the Nobel Prize. So I would expect that him and maybe there's a couple other people that were kind of involved in the whole process. So why don't you, we really haven't, in any of the previous podcasts, we haven't really talked about any of the specifics at all of optogenetics. So could you summarize that? Because it's really
this is the optogenetics is kind of defined as new technology that use light to interact with the brain. And the term comes from those experiments that Carl and others and us did at the time where Ed Boyden was also involved in this, and Pondering, where we use specific light sensitive channel or pump and target their expression to specific neurons. So that means the neurons express light sensitive channel that makes them sensitive to light. So that means that by shining light on the cell, we can either activate those cells or inhibit them. And the power and the beauty of that technology is that we can really selectively activate genetically targeted cells. So we have a spatial resolution that is that was unprecedented at the time. But we can also activate those cells with millisecond timescale flashes of light, which basically make one able to replay their true activity in the brain. With 1 millisecond, one could elicit one action potential. So we could elicit action potential and also play on the sequence and the temporal resolution of those action potential. We can ramp up, ramp down, we can do burst, you can do tonic activity, you can play with really the neural code online with light in a living brain, in an animal that is freely moving in a cage. And so therefore for us, it was very important to have access to that tool, and then to put that tool in the hypocretin neurons to show that their activity during sleep induced wakefulness is sufficient to induce wakefulness. And then since then, that technology has been used in almost all neuroscience fields.
Yeah. So the key thing here is we're talking about correlation versus cause and effect. So, for example, you see, activity, say in hippocampus, that's this pattern during REM and that pattern during slow wave sleep and so on. But that doesn't necessarily mean that it doesn't tell you really the function of that pattern of activity. And with optogenetics, you can either block that activity completely, or you can mimic that activity, say in a, in an animal, and see what happens. So
yeah, right, exactly. This is the beauty of the system, is that we can really dissect the dynamic of the circuit.
Okay, so then let's see. Yeah, digging a little deeper into the hypothalamus. So, it's a relatively small brain region, but it's important for so many things, like you mentioned, circadian rhythms and regulation of endocrine glands that produce hormones like stress hormones or other hormones. And can you talk a little bit more detail on kind of some of the different regions of the hypothalamus that seem to be important in sleep-wake cycles and so on?
Yeah, so the hypothalamus is include a lot of different nuclei that and each of those nuclei has different cell type. And although we have a good idea of which genes are expressed by those cell type, or where certain of those cells project to, or which input they receive, we still don't know completely how the hypothalamus controls sleep and wake, for instance. We know about some of the cells, but not all of them. And so as there were some very important experiment done in the early 90s, where they realized that the anterior hypothalamus contains cells that promotes sleep, while the posterior hypothalamus contains cells that promote wakefulness. And so therefore, within the hypothalamus, there are different areas, different cells that control completely opposite behavior. And interestingly, this is also true for reproduction and aggression, for instance. Same cells, same brain area there, but different cells can really control aggression or reproduction. That's quite interesting. The dichotomy between functions seems to exist for other brain function. So the anterior hypothalamus has, and since then this has been confirmed with as I said, optogenetics and many or lesions, many different approach, to the point that we know that there are many different cell type in the anterior hypothalamus that project again to wake system and also other system. When they are active, they inhibit the wake system, but they also decrease the body temperature, and then are associated with a lot of autonomic function that that accompany sleep usually, to the point that optogenetic stimulation of some of those cells in the medial hypothalamus seems to be involved in torpor or hibernation-like behavior. This has been shown. There was one study a few years ago about it.
And what transmitter, neurotransmitter, or neuropeptide are they?
So all those sleep promoting cells, they are predominantly inhibitory cells. So they use Gaba as a transmitter. They produce some neuropeptide like CCK, PV, but it's still not clear what exactly those neuropeptides do on the neurotransmission of those cells. We don't know yet. It is a little more clear though for the posterior hypothalamus with the wake system, where there's hypocretin and orexin cells. As I said, those are neuropeptide, and in those cells one can also find glutamate.
Yeah.
Activity of those cells basically release the peptide and the glutamate. And one of my collaborators, Denis Burdakov in Switzerland, has shown that the glutamate is responsible for the first excitatory response in the postsynaptic cells, so in the cell that's contacted by those hypocretin cells. And then the hypocretin kind of increase or this persistent excitation after. So for more responsible for the late response. That being said, it's a very general picture. We know that in the lateral hypothalamus, interestingly, there is another cell population that is called MCH, for melanin concentrating hormone. So this is another type of cells, not the orexin hypocretin. Those cells are inhibitory, project to many different places, but they are strongly activated during REM sleep. And if you activate them with optogenetic, then there's an increase of REM sleep as well. So really establishing a causal link between these small nuclei there and the lateral hypothalamus and those MCH cells with the onset and the prolongation of REM sleep. So, you know, just to say that although there was this difference between anterior and posterior hypothalamus, there seems to be island there also that control REM sleep for the MCH. There was another island in the dorsal part of one small nuclei of the anterior hypothalamic that contains cells that seems to be strongly activated during REM. But so far, we haven't been able to activate those ones to show that they increase REM sleep, right? It's correlation, not causation yet.
In the neurons, the GABA-ergic neurons in the lateral hypothalamus, what happens if those are stimulated?
So this is, again, another population that might be larger. It's a larger population than the hypocretin, orexin, and the MCH, but it contains the MCH as they are also GABA. And if we've been doing experiment on those inhibitory cells, the GABA cell in the LH, and we found that they are also inducing wakefulness as the hypocretin and orexin. And that's quite interesting because we believe that in amongst those cells, those inhibitory cells, some control REM sleep, right? This is the MCH cells. Some control arousal. So if you activate them, you induce non-REM to wake transition, but not REM to wake transition. So they only are important for awakening from non-REM, but don't seem to be necessary or involved in REM to wake transition. But we also know that, so they're involved in architecture, so to speak, of the sleep-wake cycle, right? But we also recently found that part of those cells are involved in food intake during wakefulness. So we knew by the past that half of those cells are active during wake and half are active during REM sleep, right? So correlation data. But the one that are active during REM sleep can be MCH, and you know, that's clear. The one that are active during wake could be controlling wakefulness, but it was not clear why they would be active between either wake or REM. And then we found that some of those cells are active when the animal is eating. When a mouse is put in an open area with food and bedding and different elements, and when the animal goes to the food pot and then eat and start eating and consume the food, we see those inhibitory cells strongly activated. And this has been reproduced by many different groups across the world. So interestingly, we follow the activity of those cells, and we found that those cells become also kind of reactivated during REM sleep. So there is this reactivation, or at least the pattern that we see when the animal is eating seems to be also present when it's in REM sleep. And that, of course, rings the bell of the reactivation of cell assemblies during sleep for consolidation, right, of behavior. And that's what we were talking at the beginning of this podcast. The interpretation of those data is that most likely those pattern of activity that are present when the animal is eating or feeding during awake behavior is reactivating during REM sleep to consolidate the behavior and make sure that this behavior is stable.
So if you activate those lateral hypothalamus GABAergic neurons, it induces feeding. That's what you said?
Yes.
What if you do the opposite? Will the animal starve?
If you silence that, they eat less, yes.
And is this, I had on a recent podcast, I had Alan Watts, who is at USC and does a lot of work on neuronal control of feeding.
Yeah.
And so do these neurons respond to leptin or ghrelin?
This is a good question. I don't know that on top of my head. I know that many cells in the lateral hypothalamus respond to leptin and ghrelin.
Yeah.
Those are the leptin receptor expressing cell, and I believe that part of those inhibitory cells have expressed that receptor. But I don't know if there was a study that really addressed the function of those leptin receptor cells. I think there is one that showed that they might be somewhat involved in postprandial level of arousal, but I'm not sure.
I guess yeah, it'd be pretty straightforward to see where they have.
I mean, I think that's where we get to read the the state of our knowledge, where we not always target the same cell, and if we want to target the inhibitory cells that are MCH, or the inhibitory cells that are leptin receptor, or the inhibitory cells that are expressing another, or cells that are connected with other brain area than the one involved in sleep, it becomes extremely challenging. It is doable. There are some tools that allow to do that, but it is where we are now. This is really what needs to be done next.
Yeah, that's an exciting time.
Yeah, are those different subcircuits, I would say, control function that at the end, are associated to each other. You can only sleep if you have sufficient metabolic status.
Yeah, it's a good situation when you have so many ideas, and you have to choose what you think is the most interesting and focus on that. Yeah. Now, let's talk about even what I think is really elegant and intriguing work. I'm trained as a developmental neurobiologist and did a lot of work on regulation of dendrite outgrowth and synaptogenesis and so on, and so was really interested in the say events going on in subregions of neurons, particularly parts of dendrites or even individual synapses, and we did a lot of calcium imaging. This is back in the 1980s when Roger Chen, it turned out he was a friend of Stan Cater, who was my mentor when I did a postdoc. And so we were among the first to do any calcium imaging in neurons. With the FIR2 back then, which is a fluorescent probe that you can introduce into all cells and look at them. But you recently, and there's now better methods to look at calcium levels, which is kind of a readout of activity in neurons in specific regions of neurons. Can you talk about the genetic tool for looking at calcium levels, and then kind of go over your recent science paper? It was I think in May of this year. Really intriguing work on looking at what's happening in different regions of neurons in the prefrontal cortex and in relation to sleep.
Yeah. So basically the tools that we have to look at calcium imaging, and you could see that as part of optogenetics, right? If you include tracking the activity of cells with light, that would be exactly that. Are usually most of the ones that we are using are GCAM. So those are mutated calcium binding protein with a fluorophore that can, when they bind to calcium, change conformation and can be excited with blue light and emit in the green spectrum. So we can measure the cellular activity through flashes of light of neurons that express those GCAMs. That's what they're called. And so we've used that to target those GCAM to pyramidal cells, so to classical cells, in the excitatory cells, in the prefrontal cortex of sleep, of mouse, sorry. And we did that because the question we had was to try to understand what generates cortical activity during sleep, and in particular REM sleep. And there are two statements there that are important. One is the one that I told you at the beginning of the podcast. REM sleep is similar, has always been considered as being a state where the brain is wide awake, to the extent that most of us still write that brain activity is similar to wakefulness during REM sleep, right? But if you look at some data in the literature, it suggests that there is very low activity in the frontal cortex, and most of the electrical signal you pick up come from axonal input, or even the underlying the hippocampus activity that generates the theta rhythm. So it was not clear where the cortical activity and what would be the cortex doing during REM sleep. And so we start imaging that, and Mattia in my lab, postdoc in my lab, who did that.
And
trained the
Antoine, from a technical standpoint, so you have the animals with their head in a fixed position under a microscope, or are they freely moving?
No, this is in head restraint preparation.
Yeah.
So
you cut a little piece of the skull out.
Yeah.
And then essentially you're putting the objective lens of the microscope.
Yeah.
Yeah.
Exactly. So that's what Mattia did first, is to train the animal to sleep in a head restraint setup. So it takes 2 weeks of slow habituation. But eventually the animal managed to sleep, and even to enter into REM sleep, very deep sleep. And as you just said, we can drill a hole and then put this glass cover slip to really have a window. It's called cranial window, to see through the glass the cells with the microscope and see the fluorescent signal of each cell. So that we can track the activity of the cells during wakefulness, non-REM, and REM sleep. And so what we found is that those cells were strongly silent. There were not much happening during REM. We saw activity during wake, a little less during non-REM, but completely silent during REM. So that was a first surprise, because we wonder where the EEG signal come from then. And so we started to image the dendrites, right? So neurons have an axon, that is the output compartment, and the input of those arborization is called dendrites. And those in the cortex are often very nice. It looks like Christmas tree. It's almost time, right? And so there are tricks to target the GCAM to those dendrites and then image the activity of the dendrite across states, same preparation, and realize that during REM sleep, the dendrites were hugely active, massively active during every single REM sleep we could recall. So it looked like we had quiescence of the cell body, but activation of the dendrite. And that basically could explain why there's high frequency activity during REM sleep that one can record with the EEG in human or in animals. That might be due to those strong synaptic input, right? So that basically suggests that there's a lot of synaptic input in the dendrite during REM, but that those synaptic input are not transformed in action potential at the output of the cell. And why is that? Is because there are interneurons. So those are local inhibitory cells that either target the dendrite or the cell body, and that their activity is very specific during REM sleep, to the point that they inhibit tonically the cell body. So there are inhibitory cells that are constantly active during REM and keep the cells silent, the cell body hyperpolarized. And the other one that control the dendrite activity are silent, and so they allow dendritic activity, and that's what really a major discovery in the lab, because we didn't expect to have this somatodendritic decoupling during REM sleep, and I mean, in a way,
So now I might forget what I was thinking if we could. So for learning and memory, there's this evidence that there's a kind of a early events occurring locally at the synapse that involve local production of new proteins, maybe, but then there's also signals sent to the cell body and to the nucleus, and there's certain genes that are turned on. You know, the classic pathway is the Krebs pathway, and so it'd be interesting to see whether, you know, you have some structural or you have some changes, I would expect, occurring in the synapses and the dendrites. And then maybe later on during the different phase of sleep or during wake, you get gene expression that follows that.
Yeah, this is one completely open question. We know that there is specific genes that are transcribed, or protein that are translated during sleep. In general, the main idea is that there's a decrease of gene expression or all this molecular machinery during sleep. That's the Caraccioli and Giulio Tononi hypothesis, that there is increase in the number of synapses and during wakefulness, and that is selectively pruned during and decrease eliminated during sleep, right?
I see.
So this is a general concept. Within this general increase, decrease, there are islands again of very specific either brain nuclei, brain circuit, or cell compartment, dendrite versus cell body or axon, that remains in which some activity remains. So there could be some gene transcription, for instance, triggered by REM sleep. This is something that is still not very much studied. It's been studied for non-REM sleep, because non-REM sleep is basically 80%, 90% of the sleep time. REM sleep is so short that it might be difficult to study mechanism that take a little more time than basically 3 minutes or 4 minutes in mice, right? But we cannot rule out that this is can be triggered during REM and then continue during non-REM. This is totally.
Is there any way using the optogenetics where you have these ion channels that they actually they're light sensitive ion channels? They actually evolved in bacteria, right? And you put them in these neurons. Is there any way to put those channels only in the dendrites or only in the cell body, and so that you can, I mean, can you mimic what you're seeing by imaging?
I mean, yes. So to answer, yes, there are tools that you can fuse some protein that basically address those ion channel to the pre or postsynaptic compartment of the cell. So this is doable. One aspect here, when we talk about the somatodendritic decoupling, would be the complexity of activating the dendrite and at the same time inhibiting the cell body to see what happens in the nucleus. It's you see the temporal, not only the spatial resolution, but the temporal constraint that we have would be very difficult. I mean, best, if there was an ideal experiment, I guess I would go for patching a cell in the brain of the animal and then suck the nuclei during REM and try to isolate that bright bit. This is something not doable, of course, but then you would have access to single cell omics approach to see what happened in this time. But yeah, I mean, there's many different aspects. Gene expression. Another aspect that I believe is quite interesting is the phosphorylation happening in certain protein at the synapses that might be involved in synaptic plasticity, both up or down scaling. that have been associated with sleep and circadian rhythm. There were two papers from two of my colleagues, and I believe this is another mechanism to which could be important, and that made me think about not close but similar mechanism where another Leo Applebaum in Israel, a good friend of mine, has shown that there is actually DNA repair during sleep, during non-REM sleep.
Oh, yeah.
So although it doesn't involve REM sleep, and REM sleep is certainly not doing all the job, non-REM sleep is very important, and such molecular mechanism can happen during non-REM here as well. So
now in this science article, there was more to it. So Mattia is Ami, is that his last name?
Amy, yeah.
And is that a postdoc or graduate student?
He's a postdoc in my lab.
Okay. So in this paper, so you've shown that there's the dissociation between the activity in the cell body and the dendrites during REM sleep in these neurons in the prefrontal cortex, and that this is due to inhibition of the cell body by GABA, certain type of GABAergic neurons, and I guess not inhibition of the dendrites. But then there's another complexity.
Yeah.
There's inputs from another brain region, the thalamus, right?
Yeah.
Yeah.
So that's what one of the questions. So I told you that basically this is somatodendritic decoupling. So that means that those inhibitory cells that exert this tonic inhibition on the soma of the pyramidal cells must be excited by something, right? And from lab, so they must receive input that drive them, that activate them. And then previous work from the lab suggested that the mediodorsal thalamus might be one player. And so we probed that brain area. So it's right below in the thalamus. And this is, again, one of these nuclei that control a lot the cortical activity in the prefrontal cortex. And so we found that there's a group of cells there that are strongly activated during REM sleep, and when they are activated, they massively project onto these inhibitory neurons. And so basically this inhibition of the cell body is driven by the thalamus, right? So and this is kind of the game now in system neuroscience. When we have an answer, when we read the calcium activity of one cell, we always want to know, okay, what drives that cell? And so we jump one synapse back all the time and try to map all those circuits. So the question is now, okay, what does activate those major dorsal thalamic cells during REM? And yeah, this is certainly an interesting question. The same question also is also true for what control the dendrite. Where are the input controlling the dendrite? So those are things we are studying now, but basically, we kind of have an idea of the basic mechanism of how this somatodendritic decoupling occurs in the brain during REM sleep. There might be other inputs, that might not be the only one in the brain. There's many recurrent connection. It's possible that other inputs are also important.
So you've talked about the hippocampus, the prefrontal cortex, and the hypothalamus and thalamus. I haven't seen, I'm not I don't follow this field very closely, but does anybody study the sensory cortexes, the visual cortex, the auditory cortex, and are they playing any role at all in the functions of sleep?
So they are involved. They show the basically the same rhythm or very similar rhythm as one we've described. Slow wave, and even in the visual cortex, there's this alpha rhythm in human that is used to define resting state network in fMRI. So that's yes, there's a lot of study focusing on the visual cortex for brain plasticity during sleep, because there's model of monocular deprivation in animals that change or modify the the structure of the visual cortex or the neural circuit in the visual cortex, and then those change in really anatomical and functional changes is an excellent model to study brain plasticity. And it's been shown that sleep is involved in this remapping of the visual field.
Oh, I see.
So for sure, the sensory cortex is less. I mean, is used. It's been strongly studied, intensively studied a while ago by Steryad and other groups, because they were an easy model to describe the dynamic of the thalamocortical during sleep. These up and down states I was talking about, they've been described in the somatosensory cortex. There is now a new era of sleep study. Jan Born in Germany has started that, and it's widely used in many labs, where auditory stimulation are used to either improve sleep or improve cognition associated with sleep. So this is another aspect that certainly involves the auditory cortex in creating this auditory evoke potential that travels and that seems to, I mean, if they are well phase locked to spontaneous slow wave in the brain, then they increase those slow wave. They might improve sleep recovery and improve memory, declarative memory consolidation.
That's true. If one were to, if you just go on, say, YouTube, and you put in sleep, a lot of the things that'll come up will be these recordings of sounds, with this certain pattern of sequence, and that's supposed to help you get to sleep. But also then that sound it keeps going on during. So is it that it's helping you get into sleep, or is it something
it's
actually?
It's not something that really put ones to sleep. It would be more something that stabilizes the sleep and kind of restore some sort of slow wave that are necessary, high amplitude slow wave, slow oscillation that are necessary for recovering sleep. We haven't talked about sleep homeostasis, but that's basically an indication of sleep pressure. The higher those slow waves are, the higher the amplitude of those waves, the higher the sleep pressure and the sleep recovery is. So that's an indication of the sleep homeostasis process in the brain.
I have another podcast scheduled with Ted Abel, who he's at I got my PhD at Iowa, and he's at Iowa, but I'm going to have one on sleep deprivation and its consequences. So maybe it'd be be a good follow-up to this.
That's a very interesting one.
Now, what about implications of your work for translation? Are there any? In other words, so now you're talking about some evidence that if you have input to your auditory system with a certain pattern of sound during sleep, it may help consolidate memories and so on. Have you thought about any implications for translation of your work to something?
Yes. Besides the sleep-dependent memory consolidation, one aspect is this sleep homeostasis, and then in another study we showed that the activity of this medial dorsal thalamus is really important to pace those slow wave and the sleep homeostatic process. So it's possible that that clearly identifies cells or not molecular target, but at least cells that are key for regulating those sleep recovery process. Now it's the goal would be to identify a specific target in those cells to be able to manipulate their activity. Another aspect also is the fact that by really defining how the brain works during sleep, I think we open a lot of doors to which tools, which methods we could use to modulate brain activity during sleep to improve. It could be mental health, stress, for instance, or PTSD. I think the science paper deal a lot with that. the idea that sleep can be used with to modulate PTSD symptom, and down to again brain plasticity. We have a study where we show that those slow wave in the cortex after stroke, right, in the remaining part of the brain that is still alive after a stroke, can be, so if we elicit slow wave with optogenetics there in that area, we promote plasticity, and then we really facilitate fine motor recovery.
Oh, interesting.
And so that really, that's a paper, a study we published in 2020, and that really also echoes some of our colleagues who do neuromodulation, TMS, for instance, modulation in stroke patients after stroke to really recover this brain plasticity during sleep with subthreshold stimulation that probably have no effect on not even on sleep, right? Because there are so few of those. It doesn't modify sleep. It's unlikely that there is a major side effect, but they promote sleep plasticity and might contribute together with other methods, right? It's not the only one. You still need pharmacology and all regular treatment, physical therapy to really, it's one other tool. Sleep here is another tool, and sleep waves to really cure brain after brain trauma.
Yeah, so you're working with neurologists at your university
Yeah.
hospital then.
Yeah, I'm next door to neurologists, and then we have a tight communication between both us, the basic, and the neurologist too.
Now, so you mentioned transcranial magnetic stimulation, TMS. How I guess how specific in terms of brain subregions can you manipulate the activity of neurons?
So with the TMS is quite a large part of the brain. It's not as specific as what we can do with optogenetics. Yet there are new technologies with radio frequencies or different type of magnetic stimulation, or that can be much more precise in controlling smaller brain area. And that is important because, of course, it allows to be much more specific to brain nuclei, maybe not circuit, but at least brain nuclei. In extreme case, I think it's always better than the tool we have right now. And ultimately, I think we may think about a way to deliver specific molecules to cells and then to excite those cells with non-invasive waves or from outside to activate those cells. I mean, this is I think there's a lot of effort.
Yeah, so in the future we may have some device we can, maybe wear at night or something, or that'll optimize our sleep and our kind of enhanced consolidation of.
There is already some headbands with auditory stimulation. So that does exist. It's available. It can most of them track your sleep with quite an impressive precision, I have to say. The auditory stimulation is something a little more difficult to do because if it's not delivered at the right phase of these waves, it can just do the opposite of what you want to achieve. So it's a little more complicated. Right now it's mainly at the experimental level, but eventually with so the machine learning and deep learning algorithm that can detect brain activity and feedback any type of signal into the brain while we sleep might maybe help us to improve some condition. Might not be a unique tool, certainly has to be associated with other more regular treatment, but it might contribute.
Yes, and
before that, I think always one thing that is important to keep in mind is that sleep is probably your best friend into trying not to get too early into those type of disease as dementia and so forth. So I think there's a higher consciousness about the importance of sleep now in society, but still we are still very sleep deprived society, and this has a major impact on the long run. I used to say we can all live without REM sleep. We just don't live in an optimized way. and then we can develop, bad mental health. We can be more stressed. You can,
Yes, and
anxiety, panic disorder, and therefore I think it's a state that is it's important to take care of that on the long run.
yeah, yes, sleep deprivation is a risk factor and associated with depression and
Cardiovascular risk, hormonal diseases,
Alzheimer's
diabetes,
disease. Yeah,
Alzheimer. Yes, it is a
it's
high association. Yeah.
right. So turn on the full spectrum light in the morning, particularly, this time of year where the daylight's getting shorter, and then, don't work too hard and don't be, exposing yourself to a screen right before you go to sleep. And maybe these auditory input, I think I might try that. I mean, I can just easy to try and see.
It's, I mean, I think there's a lot of everyone has his own strategy to promote sleep and to help with sleep. I think that's another important factor before getting to pharmacology.
Okay, Antoine, it's been great talking with you, and I hope our listeners and viewers learned a lot. And kind of the main point is there's a lot of really cutting edge research going on with sleep and trying to understand what's going on in the brain and its importance, and ultimately how we can use this information to improve brain health and general health. So thanks a lot.
It was a pleasure. Thank you very much.
Okay.